It's 10 o'clock on Wednesday, the 2nd of March 2022, and a warm welcome from U3A Helderberg, especially our non-Helderberg visitors today. I'm Richard Tompkins, your host for the presentation, which is the last of the current series, but more of that later. We have thanks to our studio director today, Jeff Burton, who is coordinating proceedings. These talks are presented via YouTube. We in the broadcast studio cannot see or hear you, but there's a live chat area on the right-hand side of your screen where questions to the speaker can be typed during the presentation. Jeff will select those of your questions that could be of general interest and show them to everyone on their screens. The full presentation today is being recorded and will be available along with previous ones on our website, u3ahelderberg.co.za. And I'd like to take this opportunity of thanking our technical team of Johan, Derek and Jeff for facilitating these recordings. Now I'd like to introduce today's speaker, University of Cape Town Professor Emeritus Anwar Mall. He hardly needs an introduction to us, but by way of background, he's a medical biochemist by training, and among many other appointments was Professor of Surgical Research in the Division of General Surgery. A year ago, we hosted Professor Mall's talk, The Complex Life of the Gut. Not an enticing title, but the information garnered from this fascinating talk precipitated calls for more of them. So a year later, we are happy to welcome him back to U3A Helderberg. His topic today is entitled Mucus and its Role in the Gut. And on that note, over to Professor Mall.
Hello. Have you got my talk on, Richard? Hello, everybody. I hope you can all hear me. Hi. Can you all hear me? Okay. Well, thanks for having me. As Richard says, I was here a year ago. So I've come back now, and I promised a talk on mucus. So it's going to be mucus and its role in the GI tract. Why does the stomach not digest itself? Next slide, please. Okay, so for anybody who has any doubts about mucus and its importance, look at the second title here. Mucus and mucins may become the medicine of the future. So this is no small stuff. And I'm very proud to say it. This is my specialty, and this is what I've done over the years. Next slide, please. Okay, so here's the whole big article. Mucus and mucins may become the medicine of the future. Next slide, please. Okay, I want to pay a tribute to this most wonderful man who's been my mentor for over 40 years, Professor Wieland Gevers. He invited me to Cape Town, to UCT, and said to me, Anwar, we're going to look at why people don't get ulcers. Is it their mucus barrier? And if so, why do they get ulcers? Does the mucus barrier break down? So that was the question. Next slide, please. Okay, so he took me on. I'm sure he had many doubts about me. I looked like a hippie then, but he was very, very kind to me. Next slide, please. So I gave a talk not long ago on mucus, slippery, sticky, slimy, but sweet and satisfying. Next slide. And that was in honor of DJ Duplessis, who was the Vice Chancellor of WITS. And it was an eponymous lecture at the Surgical Research Society, which was then published in the South African Journal of Science. Next slide. Okay, so people laugh at me. I've been called Dr. Snot, Dr. Spit, And I didn't mind. It was a very unique field that I began at UCT and in South Africa and even on the African continent. Next. And even in literature, I mean, mucus is always mentioned. Just click, Johan. Snot's not that bad. James Joyce spoke about the snot green sea. I would rather choose snot than stay in the house, says Paul Throw. The significance of snot and other effluvia in the plays of William Shakespeare. Nasal discharge is a metaphor in the fiction of Jane Austen. So we're in good company. Mucus is quite widely known. Next slide. And it was always in the traditional textbooks called an excellent lubricant. It had adherent qualities that bind it tightly to food particles. It prevents contact between food and the stomach wall. And it adheres to fecal pellets, making their passage through the gut easier. Next slide. But is it only a lubricant? The integrity of the mucus, the slimy partner in this business, barrier, is one of the first lines of protection against the harsh internal milieu of our major inner tracts of the body. And that's what I want to tell you about. Next slide. So take the respiratory tract. Sitting in your respiratory airways is a blanket of mucus sitting on these little structures called cilia, which protrude from the cells. And the cilia are motile, they move. So if you breathe in a foreign agent, it gets trapped in this mucus. The cilia beat one way and the blanket beats the other way. And so you cough the foreign agent out and prevent it from actually reaching your lungs. Now, in the cervical tract, for most of the mid-cycle, there's a plug of mucus sitting at the mouth of the cervix, not allowing any foreign agent to enter the cervix. During mid-cycle, you get the mucus changing its structure, becoming more watery, facilitating the movement of sperm up there for fertilization. And of course, sexual intercourse would be very, very, very painful if it hadn't been for the lubrication by mucus. Next slide, please. So then there are some terrible diseases associated with a hypersecretion of mucus. You don't want mucus to be in too little amounts, and you don't want it to be in too large amounts. So I spent some time in Oxford at the laboratory of Professor Anne Harris, studying mucus in cystic fibrosis. Now, those of you who are familiar with this illness, it's a terrible illness where the mucus clogs up all your passages. And you know, children used to be, the only treatment for them was physiotherapy, where the physiotherapy is to lay them down on their stomachs and beat them on the back. And their lifespan's very short. But now we're in the era of genes and gene editing. So things are improving slowly and it's looking more promising, and children who used to die early now live at least until the age of 30 or 40. Next slide. And then there's this horrible disease. This is the abdomen of a 58-year-old policeman who had this disease called pseudomyxoma peritonei. It's classified as one of the most rare cancers you can find. And you know, ladies and gentlemen, in my first 25 years of research at UCT, I saw one case of pseudomyxoma. And for the following 10 years, I saw about 20 cases. And in fact, because I published work on it, people from all over the country came to see me, and I kept reminding them, I'm not a clinician, so I can't really help you. I'm studying the disease from the point of view as a scientist. So anyway, it's usually in men a burst appendix through cancer, and in women it can originate in the ovary. But mucus begins to fill up in the abdomen. And whenever I had mucus sample, I had about 1 ml that I could get from a colon or from a stomach. But in this particular instance, I was called by a surgeon on a Saturday and said, I got some mucus for you. So I said, well, I'm not driving up for 1 mil of mucus. He says, no, I've got 6 liters for you. I didn't believe him. I did get 6 liters. They virtually vacuumed out, ladies and gentlemen. They virtually vacuumed out the mucus from his abdomen. Next slide. And this is what it looked like. Thick, like a waterfall. Next slide. Very viscous. Next slide, please. So there are other diseases. No, before that. Back one up, please. Okay, there are other diseases. In about the 80s, people began to find changes in the mucus. Now, just now, I'm going to tell you, when I talk about mucus, I talk about a total secretion. That crude gel that lines the surface, or what we say, the mucosal lining of the gut, right? But what gives it its viscosity? Its viscosity is given to it by a mucin. So please remember, mucus is the total crude secretion. Mucin is just a glycoprotein that gives this the property, gives the crude gel the property of stickiness. Next slide. And the layer is continuous across the entire gut, and across the vagina and cervix, across the respiratory airways. And there's 2 phases to it. One very, very thick and strong, the dark blue, and one a little looser. Now, you might have the impression that this lining that lines your stomach is very, very thick. It's only 200 microns thick. We're talking about a very thin lining of a gel, which offers so much protection to you after you've eaten and secreted acid and pepsin. But I'll tell you more about that. Next slide, please. So, the last time I met you, I spoke about mucus in the gut-brain axis. It has become known that without a healthy mucus barrier in the large bowel, the existence of the microbiota will be compromised. And I've spoken to you about these microbes that populate our guts. So the integrity of the mucus barrier depends on our dietary habits. Many health and disease conditions are linked to the state of mucus, the mucus barrier in the gut, respiratory, urinary, and cervical tracts. Next. So, fight them or feed them. How the intestinal mucus layer manages the gut microbiota. Oops. The intestinal tract is inhabited by a tremendous number of these microorganisms or bacteria, termed the gut microbiota. And the thick mucus layer of the gut provides a barrier to infiltration of the underlying epithelia by both the normal microbiota and other pathogens. Some members of this microbiota utilize mucin as a nutrient source. So that's very important, that even though these bacteria are our friends and they're working in our favor, they must not penetrate the mucus barrier and they must not get into the bloodstream, because then they become toxic. Next slide, please. So look at this diagram. Here you've got a beautiful sea blue mucus layer sitting on the surface of your mucosal lining of your gut, of your intestine. And look at this, sitting on top of that are the microbes, our friendly bacteria I'm talking about now, okay? And inside the mucus layer, a whole lot of protective molecules. And if you see this dark blue patch, the mucus is extremely strong. It's called the mucus colon. And it prevents the microbiota, although they're friendly, from penetrating these cells and getting into the blood. And there are also binding factors on the mucin, which gives this the gel-forming properties. There are binding factors there which help the microbes bind to aspects of the mucin to feed on it, because the mucin molecule, which imparts gel-forming properties to the mucus layer, are 80% sugar. Next slide, please. I hope you're following me. So that was, I just spoke about that, so you can go on to the next slide. I just spoke about that on the diagram. The next slide, please. Now, if you have a fiber-rich diet, you'll have a lovely, reasonably thick layer of mucus, and everything will be hunky-dory, okay? If you have a fiber-deficient diet, if you see here, it's paler than this one. So you're going to allow for penetration by organisms, not only the friendly bacteria, but other bacteria. And in diseases like colitis, I'm talking about ulcerative colitis and Crohn's disease, you get all sorts of bacteria entering your mucus layer and getting into the cells and getting into the circulation. Now you're in trouble. So always have a lot of fiber so you have a beautiful layer of mucus. Next slide, please. Now, this is Tony Caulfield. He's a friend of mine. He's a professor of mucus biochemistry at Bristol, and he's a very nice man. Every time we met at an international conference, he carried an extra bottle of shampoo, hair shampoo. I had hair then, a toothbrush and toothpaste, because he says, Mal, I know you're very forgetful. So he says that you've got these microbes in your mouth, in your stomach, and the abundance of them are in colon, and he mentions the species, which I don't need to go through here. And he says that ethnicity has been shown to influence the GI tract microflora, and this needs to be considered when comparisons between different population groups are made. Next one, please. And so, ladies and gentlemen, if you have good fiber, right, then that fiber can also convert into, so you call them prebiotic, they can convert into probiotics, okay? That fiber also elicits the formation of modulatory cytokines, which give the mucus more strength. That fiber could result through fermentation of short-chain fatty acids, and apparently, and I'm not putting my head on the block, the research is showing that these short-chain fatty acids, which are also produced by the gut bacteria, they communicate to the brain. So if you take probiotics, and if you take the right one, I'm not an expert on probiotics, then you get an improved gut barrier function, okay? And you can prevent colonization of the mucus barrier by harmful antigens or harmful bacteria. Next one, please. So today, the research is taken off. Mucins, which give you gel-forming properties, gut microbiota, and postbiotics' role in colorectal cancer. They're beginning to look at this in disease, various diseases. Next slide, please. Okay, now look at this. If you have a Western diet and other factors, you'll have dysbiosis and you'll have a poor mucus layer. If you have good that means a lot of fiber and good probiotics and a prudent diet, you'll have eubiosis, that means a well-functioning mucus layer, and you won't actually get infiltration into the bloodstream. Okay, next one. So I'm just showing you the latest research. There's another paper, mucus barrier, mucins, and gut microbiota, the expected slimy partners that actually now are part of the gut-brain axis. Next one, please. So let's talk about how this all came about, this historical background to mucus research. Next slide, please. So if you publish in the journal Nature, your career is made for life. And Nature once had a cover story. Why doesn't the stomach digest itself? It was by two people at Boston University, Ramakrishnan Bhaskar and Lynn Reed. The next slide, please. Now, even Scientific American got hold of it. Why the stomach does not digest itself. It says here the organ secretes a strong acid that can dissolve metals and kill living cells. How it avoids digesting itself and what happens when the safety mechanisms fail, I explained. So this is your stomach coming down from the esophagus and then your main body of your stomach and then going down to the duodenum. and it's covered by a thin layer of very strong crude mucus gel, right? And when your food enters here, this sphincter and that sphincter close, and then your food is battered into submission. Your piece of steak, when it leaves the stomach, it will be absolutely fluid because of hydrochloric acid and pepsin. And at the height of your digestion, if I took a drop of this acid and put it onto your skin, it will burn a hole right through your skin. And yet, the stomach wall is protected. And people ask why. Next slide, please. Why the focus on the stomach? Next one. Well, it's an organ of social adjustment. Genius, doubtless, like an army, travels on its stomach. But what is stomach? The reason that we stand in awe of the stomach is that this organ, more than any other of the viscera, controls our social adjustment. Next slide. And that's absolutely true. You'd agree with that. And this is a very conservative Muslim organ. And he says those who are unable to adopt the austere methods of eating exhorted in the hadith should at least not overeat. As the stomach is about to be filled, one should exercise restraint and stop eating. Now, if you talk about the hadith in Islam, that's the sayings of the Prophet. The Prophet advised 1400 years ago, the Prophet of Islam, Muhammad, advised don't overeat. So he probably was a gut man. Next slide, please. Just click, please. So this organ, the stomach, secretes a strong acid that can dissolve metals and kill living cells. Gastric juice can digest meat, as you've seen, as I've said to you. Then why does the stomach not digest itself? Next. Sometimes it does. And when it does, you get ulcers, terrible ulcers. Okay. Next slide. So porcelain. Why does the stomach behave as if it were made of porcelain? Asked Claude Bernard in 1855. Is it because the cells that line the wall of your stomach are so tightly placed together, even tighter than your skin cells, right? So nothing can go through. Is that the reason? Or is it the mucus on the mucosal surface of the epithelium which gives it protection? Next. And this horrible man heatily came and said, mucus, it's merely water. How is it going to protect against acid and pepsin and enzymatic digestion? And how is it going to protect the stomach wall? He says it was too watery. So this is the lumen lining of the stomach, this diagram that I showed you. And so we're asking the question is, under this mucus layer, why are these cells lining the wall not harmed in any way by this acid and pepsin? Next one, please. So whenever I opened the stomach along its greater curvature, you could see it glistening with its little bit of mucus on its surface. Next slide. And in Scientific American, if you look closer on a microscopic study, you'll see those little pits that you see on the stomach, wavy pits, are actually glands that extend right into the stomach wall. Next slide. And that stomach gland looks something like this. So the mucus secreting cells are on the top, and then below you have mucus neck cells to replace these. And then you have a parietal cell which secretes your acid. You have endocrine cells, and you have chief cells which secrete pepsin. And this is just one gland, but just to show you how complicated but beautiful it is, and how it secretes so many different components and then forms a mucus layer, and you can digest your food and not even think about it. Next slide, please. So the problem was, and you know, gut research went through many phases. I used to call them fads, but I noticed that the Americans don't like that. Okay, so now this man Schwartz in 1925, he said, no acid, no ulcer. Now, you know, people make statements like that, but if you're important, they're taken seriously. Next click, please. So he said stress and lifestyle contributed to acid secretion and induced gastric ulcers. Please note this point. Stress and lifestyle contributed to acid-induced gastric ulcers. So he recommended antacids. So you take your Rennies when you're feeling a bit bloated and uncomfortable, okay? So you neutralize your acid. But later on, they had H2 receptor antagonists. That means for acid to be secreted, this receptor on the acid-secreting parietal cell had to be stimulated. Food touches on it, and then that cell secretes acid. So if you have an antagonist against this receptor, then you can actually stop acid secretion totally. And so this is how the idea of proton pump inhibitors came into being. But unfortunately, they found that while it helped the patient with gastric ulcers, when you stopped it, the ulcers came back. Then somebody asked a very important question. Hydrochloric acid, is it such a bad guy? Is it not there for a purpose? The good Lord gave it to us. Okay, it's very strong. It can burn you. But why is it there? Because it has bactericidal and antimicrobial properties. Whatever you eat, no matter how sterile you think you're being, you're still taking in germs. So suppression of acid secretion for too long a period can be problematic. In my gastric cancer patients, there was obviously no acid in their stomachs. Next one, please. So this man then came up with the new idea of cytoprotection. He also asked, he said, no, I think it's the architecture of the stomach wall and the wall of the gut. And that architecture of those cells is different from the arrangement of cells anywhere else in your body. And so he coined the phrase cytoprotection. I heard him speak in New Orleans. He was a very smooth speaker. Next slide, carry on. And he says cytoprotection is strengthening of the stomach surface epithelium, right? And so he said, let's focus on strengthening the cell wall and not inhibiting acid secretion, because acid secretion nature has given us for a purpose, okay? So let's enhance the stomach's own inherent mucosal defense mechanisms. Next one, please. So, cytoprotection by prostaglandins. So they started feeding people with prostaglandins, which are little fatty acid molecules. And they improved the circulation in the stomach tissue. They tightened the gastric mucosal barrier. They stimulated mucus secretion. And they became a blessing for rheumatoid arthritis patients who got ulcers because they were on anti-inflammatories. So if they took a prostaglandin before taking the Indocid or indomethacin or whatever, they didn't get ulceration and they were very happy. So it was successful for a while. But, next slide, please. But the problem was that prostaglandins have side effects. Prostaglandins are used during difficult labor. So they had side effects. So now a new hypothesis came. Perhaps mucus has some role to play in the protection of epithelial surfaces. Let's revisit it. It has to be playing some role. Let's go. Continue. So mucus biochemistry was born. Next slide, please. So then they started formulating. They moved away from H2 receptor antagonists like cimetidine and ranitidine and started using non-anti-secretory drugs that will not interfere with your acid secretion and with your mucus secretion. Drugs like carbenoxolone, sucralfate, a viscous paste, denol, a bismuth compound. All the above promote healing and do not interfere with the secretion of acid. But they haven't been 100% successful. But at least you still had no interference with stomach physiology. Next one, please. What they did is reinforce the mucus barrier. And then these two guys came along in the 80s and said, do you know what? Ulcers are formed by a germ called Helicobacter pylori. And they won the Nobel Prize for it. And they went to great lengths to actually prove that these ulcers are caused by these germs. I was in Sydney when they made this announcement. And this man, I think his name is Warren Marshall, he drank a whole glass full of Helicobacter. And apparently his ulcer still hasn't cleared. Next one, please. And so I went and looked into one of my patients to show how these germs sit there. These little ones, they sit in clumps. They're called Helicobacter pylori. And this is what apparently causes damage to the mucus. Next slide, please. And so a new model for protection was born, that your lining of your stomach, right, or wherever, then has a nice layer of mucus sitting there, okay? And then you've got acid and pepsin in the stomach, which keeps attacking the mucin molecule. So you've got the mucus gel. Now you've got a mucus gel which is made by mucin and other factors, but the mucin gives it its sticky properties. So it sits there on that. And it gets degraded by the enzymes in the stomach, but as it gets degraded, more mucus is secreted. Okay, and the activity of the stomach when digestion takes place is like it's like a washing machine, but you don't feel it because as abrasion thins out this mucus, more mucus is produced. So it's a fixed layer, and under it has some bicarb. So if any acid does penetrate the mucus layer, the bicarb neutralizes it. Okay, so that's the model for secretion. And below the mucosa then is the blood vessels. So things don't actually go through and destroy your cells. Next slide, please. And so here we took some live tissue and we looked at it. And when you do have an ulcer, then the mucus and the fibrin, which actually clots the bleeding, form an extra strong barrier. And then new cells are able to come in and heal your mucosa. Next slide, please. So the gel-forming mucins give mucus its sticky property. I've said that a few times, and I want that to actually sink into you. Mucins or mucus glycoprotein only make up 1 to 5% of this total secretion, but it imparts the gel-forming properties, the stickiness, so the mucus sticks there. Next slide, please. Now, when I go to theatre, I used to open the stomach around the greater curvature, open the colon, or open the lung, and scrape off the mucus with a glass slide. Okay, next slide. Now, its main components are too many. You can have dead cells, you can have protein, digested food, bacteria, lipids, but its stickiness is due to the mucin, which forms 1 to 5% of the total secretion. And this mucin molecule, what is it? How is it that it's so fantastic that it gives these gels their sticky property? Next slide, please. If you asked me to explain to you what a basic unit of mucin looks like, it looks like a bottle brush. So the wire is the protein part, and these bristles are the sugars attached to it. It's 80% sugar. Okay, and what we say is that because it's a sugar that's glyco, and because we have some protein here to which the sugar is attached, we call it a glycoprotein. Okay, next slide. Now please remember the bottle brush structure. Now this is one bristle. Look how many sugars it has attached to it. So it's a long sugar chain attached to it. So another way of mucins, which form gels, protect the gastric mucosa or the lining or the cells of the stomach, is that it's got so many of the sugars nothing can penetrate to, and if some bacteria do penetrate, they get caught by these sugars. Next slide, please. And that's very important. So I spent 3 happy years with a team of researchers, mucus researchers in Newcastle upon Tyne. This was Professor Adrian Allen, who was the head of our department. And these were all my colleagues. They're spread all over the world now. I'm in South Africa, as you know. Okay, next slide, please. And Adrian Allen formulated a structure for this mucus. Now, please just bear with me. I got to explain this to you so you understand how we can then use it to understand disease. So, these are the bottle brushes I showed you, the glycoproteins. Each one you can call a monomer, but when they link up in this windmill conformation or structure, you call it a polymer. If you break this, then you go back to monomer. What is very important, Adrian Allen always stressed, for you to have a good mucus gel, this must be polymeric. All its subunits or monomers must be together. All the bottle brushes must be together. And then you get polymers, and this polymer will then join with another polymer, which will join with another polymer, which will join and form a gel. When pepsin attacks the mucus barrier on the surface of the mucosa, it breaks this into the monomer. And when it breaks it into the monomer, if you get more monomer or subunit instead of unit, you're going to weaken your gel. I hope you got that. I'm prepared to, at question time, repeat that. Next one, please. So, wrong, said John Sheehan and David Thornton, that the bottle brush structure basically is correct. John Sheehan is late. He was in Manchester. And Dave Thornton is now running his lab. Two very good biochemists. Next one, please. They said, yes, sure, we have bottle brush structures, but it's not a windmill structure. It's just straight chain random. And the polymer attaches to another polymer, just like Allen proposed. And if you break it up, you get monomers or subunits, and you weaken the gel. So basically, they agreed with each other about everything except the structure. So biochemically, we actually failed to determine whether it's a windmill conformation or a random coil structure. Next slide, please. And then the world of genes descended upon us, genetics. And when they looked at the gene for a bottle brush, they found it to be a very complicated structure. It had a whole lot of other proteins attached to the mucin, and that itself is pro von Willebrand factor. That comes from the blood, and that is attached to your mucin, and then there's another protein here, and so it's extremely complicated. So to actually wonder now whether this organizes a monomer into polymers and windmill conformations and random coils or straight chains is a moot question. But what we are doing is we are understanding that it's a very complicated structure. The simple bottle brush is a very complicated structure. Next slide, please. And so that gene codes for the wire part of this, and then the sugars are added onto it, and you get a monomer, which joins to form polymers, and polymers join to form gels. That's the basis of the story. Next slide, please. So here's your model again. For there to be a viscoelastic, strong gel on the surface of the cells of the mucosa, you must have more polymer and less subunit. So joined bottle brushes and less free bottle brushes. Then you get a good gel. If acid or pepsin break the mucin polymer into subunits, then more mucus is produced, more mucins are produced, and this gel stays consistent as 200 microns thick. Next cell, please. Next slide. So in certain diseases, how are mucus and gel-forming mucins associated with disease? The mucin polymer forms the gel. Sorry. The mucin polymer forms the gel, and there's a dynamic balance on the mucosal surface, right? If subunits suddenly increase in relation to polymer, you get a weaker gel. Next slide, please. So in ulcers, polymers are too little. In cancer and cystic fibrosis, polymers are in excess. Next slide, please. So when we tested it, we took some pig gastric mucus and we found that it's got lots of polymer. And when we broke that polymer up, we got subunit or just plain single bottle brushes. When we looked at duodenal ulceration, we found that there was more subunit than polymer, weaker gel. And when we looked at a patient who had a gastric ulcer, again, lots of polymer in relation to, lots of subunit in relation to the polymer. Next slide, please. And I did some work in a gastric ulcer model where you had a nice mucosa at the beginning. And then after that, we cut off the bile duct of this model of a pig, and then it began to erode, and it eroded down, the mucosa eroded down to nothing. Guess what happened? While these changes were taking place, next slide, the mucin levels dropped from normal to pre-ulcer to ulcer and to post-ulcer. So the mucins were related to keeping the mucosa protected. Okay, next slide, please. I looked at gastric cancer in the Western Cape. At that time, in those days, we had 69.9 per 100,000, which was considered to be a very large number. Next slide, next one. Are the mucins any different in these individuals? We published this work. Yes, they're different. Next, they're actually more broken down in the cancers. And then there was a horrible kind of cancer called Menetrier disease, where the surface of the stomach looks like the surface of the brain. That too had very, very damaged mucus. Next one. And there we found a lot of small mucin stuff, more than big mucin stuff. So mucus was actually implicated in gastric cancer and gastric ulceration. Next slide, please. And if you looked at the normal stomach under the microscope, it looks beautiful. Once you start getting changes like intestinal metaplasia, you see the architecture suddenly breaking up. And in gastric cancer, you just get an increased expression of that little protein that I showed you. Next one, please. And this work was done by Dr. Tato Chirwa, one of my PhD students. Next slide. And Tato did very well with us. He identified this protein that appeared in gastric cancer. He called it alpha acid 1 glycoprotein. He won the Bunny Angon Prize, and he went and spent a few months in England as part of that prize. Next slide, please. So the lesson, ladies and gentlemen, that in science there are no assumptions. Next slide. Always provide evidence for what you are saying. And then what came along was in the 1990s was a new kind of mucin, looking very much like a bottle brush, but this wasn't a gel forming one. This wasn't secreted from the cell. This was actually attached to the cell. And we didn't think it was terribly important. It stuck out like a TV aerial from a roof. That's your cell down there. But we later learned that it's extremely, extremely important for disease. Next slide, please. Okay, so in a normal cell, you have apical polarity. That means these sit at the apical surface, right? Stretching out into the lumen like TV antennae, and that's your cell. When your cell becomes cancerous, this multiplies and this surrounds the entire cell. Surrounds the entire cell. So the theory is that now this cancer cell, transformed cell, and this is in breast cancer, this transformed cell wants to move and metastasize. It wants to go to your lymph nodes. It wants to go all over your body. What this does is it dodges the immune system by shedding these MUC1s, these abnormal MUC1s. And once they shed, your immune cells are diverted to attacking the shed ones, while the cell very cleverly and stealthily moves through the bloodstream and goes and colonizes other parts of your body. That's how cancers metastasize. Next slide, please. And so this is MUC1 in cancer, and I told you about shedding, and I told you about evading immune surveillance. Next slide. So if you look at a cancer MUC1, there's an aberrant glycosylation and aberrant sugar branches, very brief, and the mucins are very vulnerable. This protein core can be broken by enzymes at any time. In the normal state, this protein core is very protected. Next slide, please. And then we looked at HIV/AIDS. The question we asked is, why is the human immunodeficiency virus not transmitted through the exchange of oral fluids? Next slide. And then we had Hapten Hapte, a brilliant PhD student who did his PhD with me, and then I sent him to America, and then he sought refugee status, because if he was sent back to Eritrea, he would have been in trouble. Next slide, please. He found that saliva prevents the transmission. So you can exchange salivary fluid with somebody who's got HIV, and you'd be protected because of the mucus in your mouth, the salivary mucus. And I used to present this work at the symposia in the Netherlands. Next one, next slide. So what we eventually found, and just give me the next slide, please. The next slide. I want to summarize the whole thing. Next. As you can see, we published this work. And next slide. Okay, again, next slide. Next slide. I've got a summary slide. Okay, just before that, sorry. The summary was that salivary mucus, right, inhibited HIV-1. Cervical mucins inhibited HIV-1, but the crude mucins did not, right? Crude breast milk didn't inhibit the HIV-1, but the purified mucins of breast milk also. So what is going on? So if an HIV positive mother breastfeeds her child, that child is protected by his salivary mucus against the HIV. Then, if he swallows the milk, there's gastric acid. So we do get mother-to-child transmission, but we don't get it to the extent we would have had it if there was no mucus. Okay, so why do some children get HIV? And that's the question. Is there something wrong with the mucus? Next slide, please. And this is my latest findings by Dr. Baxolele Mhlukude, who's now working at Charity Hospital in Germany, is that crude cervical mucus plugs inhibit HIV-1. Next slide, please. So, and I spoke to you about breast milk. The next slide, please. Next slide. And I've given you a summary. So salivary mucins and crude saliva protect against HIV infection. Breast milk mucins, but not crude milk. And cervical mucins, but not crude cervical secretions, right? Okay, next slide, please. And then we had Julia Peacock and Yolanda Ntembu, brilliant students. Julia Peacock had finished her master's. She had a distinction for it. And she was going to do a PhD with me, and then she left me to go and get married, this silly young lady. And Yolanda Ntembu then went on to do her PhD in Gothenburg. Next slide, please. Then we've done a lot of work in asthma and other respiratory diseases. There's Ureshni Govender. Next one. Next slide. Astrid Trimel, who's doing very well at UCT, and Refilwe Mofokeng, who I think has joined the CSIR in Pretoria. She got a brilliant PhD with me. Next slide. And then I had this lovely team. Not all of them are here, and they were ever changing. But generally, it was a small group, but very, very Very, very nice people to work with. Next slide. And when I started mucus research, I had to go and produce my ideas to the great Professor Solly Marks, the head of gastroenterology at Huddersfield Hospital. And Solly Marks had his spectacles over his nose, and he looked at me, and I was so nervous. And he's known to be a man who didn't pull his punches. So Solly listened to me, and then when I was finished, I was so relieved. And he looked at me over his spectacles and he says, "My boy," I thought he's going to say congratulations. He said, "My boy, you'll come to a sticky end just like your bloody mucus." But it didn't happen. I didn't come to a sticky end. I worked very hard, and I traveled a lot with, I think this picture was taken, I traveled a lot with him to overseas conferences, to Hong Kong and Brisbane and Sydney and Miami and all over the world. Next slide. Thank you very much, and I hope you all heard me. If there's any questions, I'd love to take them. Thank you. You know, do probiotic supplements like Probiflora actually work to improve the mucus layer? You know, people feel they do, but the evidence-based research on probiotics is small. I go to the pharmacy and I see a whole section on probiotics for the gut, for gut protection. And I always tell the pharmacist, I yet have to read and see more work being done to prove this. Next question. Are there any other questions? If not, thank you very much. I hope you enjoyed that and heard me clearly.
Well, that seems to have been the last question, so concludes today's presentation. Thank you very much, Professor Mall, giving us some of your time today. Such an informative and interesting talk. I'm sure many people will now be rethinking lifestyle and attitudes towards food and digestion. We only have ourselves to blame.
Richard,
yes, sorry?
Sorry? Yeah, carry on.
As I said last month, our monthly meetings will return in person to the Strand Town Hall from Wednesday, April the 6th at 10 a.m, the first time possible since March 2020, now seeming a long time ago. The speaker will be Una Fulyun, a Somerset West resident, about her travels on the Silk Road and Samarkand. There will, of course, be a tea table offering refreshments and a biscuit for sale. And yes, I did check there won't be stale leftovers from our evacuation 2 years ago. The committee is very much looking forward to seeing you all there, and don't forget your 2022 membership badges. Visitors will be welcome. Until then, goodbye from U3A Helderberg.